And right, we are officially recording. So we'll go back, and I've said all I'm going to about Cnidaria, but there's a couple of other phyla that need to be covered. Okay, we have somebody joined. Okay, Jamaica's back. So we'll cut over to here. Okay, there's actually one last group of Cnidaria that I didn't mention yet, and that's called the Myxozoa. And for a very long time, they were thought to be protists. They were thought to be their own lineage of parasitic protists. They're obligate parasites. What we now know is that they are extremely reduced animals, probably Cnidaria, probably Hydrozoa, that have shifted to a parasitic lifestyle and stripped down their anatomy so much that you can hardly recognize that that's what they were. I won't go into all of the details of the life cycle, but many of them alternate between fish and invertebrates, such as segmented worms. The life cycle alternates between hosts, and the stage that comes out of the invertebrate will actually inject what are called polar filaments into the tissues of a fish host and pierce the cells of the fish host. And those polar filaments are basically modified nematocysts. So the body of these Cnidaria has shrunk away to only a small cluster of cells, but there are still some nematocysts that are retained and used to pierce the cells of the host. There's one species that causes a disease called whirling disease of salmonids, salmon and trout. And that's actually a problem not just for wild fish, but for farmed fish as well. There's another one that causes salmon disease called PKD. And you may not particularly have warm, fuzzy feelings towards salmon, but bear in mind that this is a multi-million-dollar industry because in many parts of the world, salmon are commercially farmed, in addition to the wild ones that are still caught and exploited for food. So this has some economic concerns. There you've got these infective spores of a myxozoan, and you can see that each spore has a couple of things that look like pears. Those are the polar capsules, and they're modified nematocysts. They're shaped like nematocysts, and each one contains a coiled filament. And these things will fire and attach the spore to the fish host. Heavy infection can cause these nasty cysts. That's a Japanese carp with cysts caused by Myxobolus naganensis. They can get into the brain and mess with the fish behavior. They can. There's one that just causes the tissues to kind of liquefy, which of course, it renders the flesh unsaleable because nobody wants to eat, salmon that has been partially liquefied by a parasite infection because ick. They can also cause damage to the skeletal tissues. They can cause deformities when they distort cartilage and bones. So this is one that's infected with Myxobolus. That's a rainbow trout with severe deformities caused by whirling disease. Fish with deformities like this can't swim straight, which is why it's called whirling disease. They'll often end up swimming in circles. And you can see the effect that this has had. So, yeah, just yet another case of very unusual Cnidarian adaptations for a parasitic lifestyle in which they may go through a stage that's as small as maybe 4 cells or something like that. They've almost completely ditched this multicellularity business, but they've still got nematocysts, and DNA sequences still suggest that they are, in fact, very weird Cnidarians, very highly stripped down to almost nothing. Okay, so the Myxozoa is the real end of the Cnidaria. That's the last of the Cnidaria we'll be talking about. There's two more phyla that are among the earliest to branch off of the animal tree of life. The first is the Placozoa. They're the simplest of all free-living animals, although those Myxozoa may be stripped down to contain even fewer cells, but they're obligate parasites. Placozoans live on their own, and a single placozoan may consist of maybe a few thousand cells, but they're grouped together in only 4 different cell types. So very little cell differentiation. There's seaweeds that have more cell types than this. And some recent work with studying DNA places placozoans as the most basal animal group of all of them. This is way down at the bottom of the animal tree of life. The outer cell layer is stuck together by these cell adhesion structures called desmosomes. It's not quite a true tissue because you don't have a basement lamina of collagen 4. And there's only one known species called Trichoplax adherens. The first ones were actually discovered crawling around in an aquarium. Because these guys are roughly millimeter size, it's very hard to observe them in the wild. We know that it's found in warm oceans all around the world. There are probably more species than just the one, because two Trichoplax might be genetically distinct enough to be separate species, even if they look identical, but very few people have actually gotten around to studying these guys. Again, they were discovered in 1880 in an aquarium. They're not easy to observe in the wild because they're millimeter-sized little blobs creeping around. They're basically flat discs. They can feed on algae by kind of humping up over a food item and digesting using the lower surface. They can glide on cilia. They've got no fixed shape, though. They can change shape to some extent. We know that they have sex, but we don't know how the embryos develop. And we know they can also reproduce by budding or fission. So there's what they look like. Up at the top, that's one that's been stained with a fluorescent stain to bring out the nuclei. Down at the bottom, that's what a live one looks like. The diameter is roughly 0.5 millimeter. That bottom picture was actually sent to me by an old friend of mine in grad school named Alan Collins, who's one of the few people who's actually worked with living Trichoplax. There's not many people. If you're looking for career advice, it would not be that difficult to become the world expert in Trichoplax. There's not an awful lot of competition. I didn't say it was good career advice. If you look at one of these in cross-section, on both sides you have ciliated cells. There's large ciliated cells on top and then smaller ciliated cells on the bottom. On the bottom side, in between the ciliated cells, you've got gland cells for secreting enzymes and mucus. And then in between, a layer of fiber cells that actually form a syncytium. And you should remember this word because it's going to come back. A syncytium is what you get when you take either one cell that divides its nucleus but doesn't divide its cytoplasm. So you end up with a cell with many nuclei, or sometimes you get a syncytium when distinct cells fuse. Either way, it's what you have when you have one giant cell with many nuclei, but no partitions between the nuclei. That's a syncytium. We've seen asexual reproduction. At the lower left, there's one that's just splitting into two. We've actually found sexual reproduction as well. And down at the lower right, there's a couple of embryos. The one down there at the lower left, number F, is at 4-cell, and then I guess that's 8-cell over at G. We have not successfully raised these in the laboratory, so we don't know how we get from those simple embryos to the adult structure, which would be really neat if we could figure that out, because as we'll see, you can get some interesting clues to how animals evolved by looking at the ways that they develop from embryos. It certainly doesn't tell you everything, but it's an interesting source of data. Back about 12 years ago, they sequenced the entire Trichoplax gene. Molecular analysis based on whole genomes suggests that Trichoplax branched off after the sponges. Down at the bottom of there, you see Amphimedon queenslandica. That's a sponge. Up there at the top, the outgroups are Saccharomyces, which is yeast. That's a fungus. And then Monocyga brevicalis is a choanoflagellate. And then everything else is a member of the animal kingdom. Hydra magnapalpellata and Nematostella vectensis are two cnidarians. Hydra is a hydrozoan, Nematostella is an anthozoan, a small anemone. So there's the cnidaria. The other animals are represented by Homo, which is us, Ladia, which is a limpet, a snail, and Drosophila, the fruit fly. And then at the bottom, Trichoplax, you've just seen, and Amphimedon is a sponge. So some analyses suggest that Trichoplax branched off after the sponges, but before the split between cnidarians and all other animals. They fit in between sponges and cnidarians plus everybody else. Right, that's Trichoplax. There's some debate on the matter, though, and currently the joker in the deck that we don't really know what to do with is a phylum called the Ctenophora. They're all marine. There are between 150 and 200 known species. And they have 8-fold radial symmetry, right? There's 8 angles you can turn a cnidarian through to get the same figure. Right, 45, 90, 135, 180, 225, 270, 3, God, I'm tired. The other one, and then 360. Sorry, starting to run low on caffeine. In many species, you have this 8-fold radial symmetry like a stop sign. In others, you've got 8-fold radial symmetry that looks like somebody squished the stop sign, and you have this biradial symmetry. I'll show you examples of that in a bit. There's a few that creep on the bottom, but almost all of them are planktonic, and they swim by means of rows of cilia. There's 8 of them, and along each row, you'll have bundles of cilia that are fused into a structure called a comb plate. It's a bunch of tightly packed cilia that all beat together. I don't know if you can see what I'm doing here, but I'm using my fingers to imitate what a comb plate does. And it's the beating of these rows of comb plates that move a ctenophore. They're the largest organisms that move entirely by cilia. Many can capture prey with a pair of tentacles, and some of them have sticky prey capturing cells called colloblasts. Collo, it means glue. So colloblasts are glue cells, but they don't actually have true nidae. There was one species that does use nidae, and it used to be thought that this meant that ctenophores were very closely related to cnidarians. But it turns out that this particular ctenophore actually eats jellyfish and steals its stings and retains them in its own tissues, but it doesn't make its own. So we now don't think that ctenophores are particularly that close to cnidarians, although they both branch off somewhere close to the base of the animal tree. Ctenophore, by the way, means bearers of combs. Ktenos is Greek for a comb, thing you make your hair straight with, a comb. So there's one. This one is completely transparent, and it's plankton. It's capable of swimming, but it's not a strong enough swimmer that it can control exactly where it's going, so we call it plankton. The mouth is actually up at the top of the picture. Extending down below it, you have a gut. In this case, it's pretty simple and tube-shaped. The body surrounding it is kind of lyre-shaped, and you can see 8 rows of little things that look like dashes. They are variously colored. This is not structural color, this is iridescence. The light is passing through this critter in such a way that when it hits the comb rows, it refracts, like light passing through a prism. And you can count 4 on the left and 4 on the right of these rows. And each one of those rows is made up of those things that look like dashes. Those are the comb rows, each of which is made up of multiple cilia, all of them beating together. So yeah, it's basically a swimming mouth. Up at the top, to the left and the right of the mouth, they're kind of coiled up right now, but there's a pair of lobes that can flex to bring prey in towards the mouth. That's how this particular one processes food. There's a nerve net. There's a special balance organ called a statocyst. There's a mouth. There's also anal pores. This is the first critter we've talked about where the way out is not the way in. The gut sometimes branches into canals, although it doesn't in the picture you just saw. There's no gut canals there. The cell layers include an ectoderm on the outside, an endoderm on the inside, and a third layer between the two called a mesoderm. And this is something that we haven't seen yet, the building of an animal body out of 3 different layers of cells. And we're going to talk about that probably starting Friday, definitely on Monday. We've got to introduce that. So there's a diagram of a fairly typical one. Pluribranchia is affectionately known as the sea gooseberry. You can see 4 of the 8 comb rows. This particular one does have tentacles, and they can be retracted into tentacle sheaths or extended, trailing along behind the ctenophore and again forming a sort of drift net-like structure. Again, very low energy way of trapping prey. There's a mouth up there at the front, and down where it says aboral organ, there are anal pores surrounding that. This is what one looks like when you kind of look towards the anal pore. You can see the diagram shows you can see 4 of those comb rows. The other 4 have been are partially visible, but have been kind of removed so that you can see some of the canals coming off the gut. And then at the aboral end, this thing called a statolith. Lots of critters have balance organs that are basically built on the principle of putting a golf ball in a beach ball. Right? Imagine you had a small hard ball like a golf ball inside a larger ball like a beach ball. Whenever you tip the beach ball, the golf ball is going to roll down, right? The golf ball is always going to be drawn to the lowest point of the beach ball by gravity. Balance organs are typically built the same way in that you've got a hollow sphere, the beach ball, with a solid sphere, like the golf ball, on the inside. And whenever the whole thing tips, the golf ball rolls to the bottom, right? Well, here you've got the golf ball is called a statolith inside a ciliary dome. That's the beach ball. And the statolith is mounted on these little nerve organs called balancers. And whenever the ctenophore tips, the statolith falls onto one of the balancers, which triggers nerve impulses that will cause the ctenophore to right itself. Your balance organs work in a very similar way, by the way. So do fish balance organs, and so on. Ctenophore tentacles are packed with these colloblasts, which shoot out whole bundles of threads that have sticky granules at the tips. There's an unfired one up there at the top right, loaded with these sticky granules that are all connected to each other on a thread. So if something gets trapped, it gets hit with a whole bunch of sticky granules and tangled up like it's in a net. And then there's a fairly typical ctenophore in life, the so-called sea gooseberry, swimming along and trailing those tentacles behind it like a long drift net. So that's what they look like when they're swimming. There's a close-up of one. Again, the rainbow colors are not bioluminescence. Pluribranchia is completely transparent, but if you get the light shining through it at exactly the right angle, the light is refracted by the transparent body, and you get this prismatic effect. If you can get the light on these just right, they're quite beautiful. There's some really nice footage of ctenophores that are swimming that I'll go ahead and include on the playlist, and you really need to see it. I've seen these guys live before. There are actually some aquariums that have them live in the same kind of tank that they would use for jellyfish. I think I've seen them at the Aquarium of the Americas in New Orleans. I know I saw them at the Estuarium in Mobile, Alabama. I'm sure there's others that have them. And they're really quite striking. But anyway, this picture, you can see the mouth at one end over at the far right, and you can see the 8 comb rows. And the footage that I'll show you will show you this beautiful iridescent effect that you get when the comb rows are all beating. When those little plates of cilia, the comb plates, are swinging back and forth. The effect's hypnotic. Right. As I mentioned, there's a few ctenophores that steal the cnidae from jellyfish. This is one. This is called Hekelia. Because of this, ctenophores and cnidarians used to be grouped into one common phylum, and the phylum was called the Coelenterata, which you might see if you read older books, Coelenterata. But we now don't think they're all that closely related to each other, and those shared cnidae don't result from common ancestry. They result from some ctenophores stealing cnidae and using them in their own tissues. Still a neat trick. This is one you commonly see in the Atlantic and Gulf of Mexico, off the coast of Florida. I don't know about the Bahamas, but I know you get them in places like coastal North Carolina. This is the so-called sea walnut, Menemiopsis ladii. This is one that doesn't have tentacles, unlike Pluribranchia that you saw a minute ago. But they handle food with a pair of preoral lobes. In both of these pictures, the mouth is up at the top. Over on the left, the lobes are kind of folded in, but there on the bottom, the lobes are expanded, swinging out like wings. And if they contact prey, they can swing in and push the prey towards the mouth. Menemiopsis is native to the North and South Atlantic coasts. It can tolerate a very wide range of salinities, so it can get up into estuaries and bays. It feeds on zooplankton, little floating shrimp, larval fish, and fish eggs. There are some natural predators that keep the population down. Jellyfish will eat them. There's other ctenophores that eat them. There's a particular fish species called the butterfish that will eat them quite happily. And in their natural habitat, the populations can generally be kept under control. Menemiopsis ladii got introduced into the Black Sea, which is in southern Russia, and it also borders other countries like Turkey. Was introduced in the early 1980s, probably in ballast water from a ship. You know, ships will pump water into their tanks to affect how they ride in the water. That's called ballast. And, you know, so they may sail across an ocean and then pump that water out. And sometimes there are marine critters that may survive the trip and end up getting introduced to a new habitat. And peak populations of M. ladii reach 400 individuals per cubic meter. That's roughly 10 per cubic foot. Imagine a box in front of your face, a foot on every side, with 10 swimming Menemiopsis ladii up to an inch long or more. Now imagine that filling up the entire Black Sea. By 1990, there was an estimated billion tons of Menemiopsis ladii in the Black Sea, and they ate every damn thing in the Black Sea, in particular fish eggs and fish larvae, which were actually commercially fished. You know, those eggs and larvae would grow into commercially valuable fish species like anchovies. But those fisheries were put out of commission by Menemiopsis ladii, spreading through the Black Sea with no natural predators. From there, it seems to have spread to the Caspian Sea, to the Mediterranean, most recently up to the Baltic. So there's your map of the Black Sea. This is a slightly older map. That peninsula in the Ukraine is the Crimean Peninsula, and Russia actually took that back a few years ago. So yeah, the map's a little bit old. But 6 countries border it, and they all have had their fishing industries badly damaged. Turkey, Georgia, Bulgaria, Romania, Ukraine, and Russia. Okay, I just heard something go ding, so... Okay, I don't see anything. I don't see anything new on the comm. So we'll go back to the slides. So yeah, I need to update that slide because Russia now has the Crimea. That should be colored yellow. Kind of a political issue, but anyway. Menemiopsis is spread from there to the Caspian, Baltic, Mediterranean, and it caused economic hardship in, you know, Russia, South Russia, Turkey, this part of the world. Now, as they say, fight fire with fire, or as Metallica said, fight fire with fire, or set a thief to catch a thief, and set a ctenophore to catch a ctenophore. A ctenophore called Boroi ovata, which looks like that, was accidentally introduced into the Black Sea in 1999. Boroi happens to be a major predator of Menemiopsis. And Boroi, together with population declines caused by exhaustion of the food supply, seems to have stabilized Menemiopsis in the Black Sea. It's not quite the problem it used to be. Sorry, I forgot I did those sound effects. So yeah, Boroi is basically Pac-Man. It basically consists of a giant swimming mouth, and that's about it. And it loves feeding on Menemiopsis. So Boroi doesn't have any tentacles. This is one that is biradial. It looks like one of those sea gooseberries that we saw earlier, but it's flattened in one direction. The mouth is on the left-hand side, and it's, you know, crescent-shaped and very wide, and kept sealed during normal swimming. But as Boroi approaches its prey, it starts opening up its mouth. There's a Boroi coming at you with mouth open. And there's one that's fully opened its mouth. And there's actually modified clusters of cilia that act as teeth on that mouth. And Boroi just bites the heck out of its prey and swallows it in great big lumps. So there's Boroi with a mouth open. There's Burrowie on the left that is just going waka, 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 chomp, chomp, chomp on a very shredded Menemiopsis ladii over there at the right. So there's Burrowie eating Menemiopsis. And yeah, there is a very full Burrowie that has just chomped a bunch of Menemiopsis and is looking very satisfied. By the way, if you look carefully, you can see the comb rows, and as often happens, they're iridescent in this lighting. In between the comb rows, you can see some things that look a little like brown squiggles. Burrowie is one of those that has a complex branching gut. The gut is central, and then there's a bunch of branches leading off from it that convey digested food to all parts of the body. So that's what you're seeing there. There's a few cnidarians that do creep. This is one, Coeloplana, highly modified for benthic life. And then my personal favorite, one of the weirdest, is called Venus's girdle, Cestum veneris. And this is what happens if you take a ctenophore and you roll it out with a rolling pin. The mouth is in the center of the belt, and so is the anus. The mouth is in the center. The anus is at the opposite end. And the comb rows extend along the edges of a body that's been rolled out up to 3 feet long. So, yeah, this thing looks like a giant transparent, almost invisible ribbon undulating its way through the ocean. So there's a couple of them. You can think of Cestum as this Burrowie-like ctenophore that's been rolled out into a band up to 1.5 meters. Okay, that's up to roughly 5 feet long. So, yeah, that is a big and very strange ctenophore. Very difficult to study. We can't keep them alive in aquariums. You have to go out into the blue water to find them. They're not the kind of thing that stays close to the beach, and extremely fragile, very hard to collect intact. So not very well known. But yeah, just a reminder that there's no shortage of mysteries lurking in the ocean. Okay, I tell you what, we are ending early because we've got another person looking at the plumbing who is going to be by pretty soon. So I have to end this, and I still owe you guys a lab. Tomorrow we'll meet at the usual time. I'll take you through some slides, and then in the afternoon I'll post the footage for you to be doing, and I'll get that third lab done as fast as I can, as soon as I can get a fix for the problems I'm having with my laptop. For those who got their microscope and slides late, you've still got several days to do the first couple of labs. I'm not going to hold you responsible for that. And one way or another, we'll catch up. So peace out. You take it easy. Have a good day. I'll post the lecture slides as soon as I can, and I'm going to stop recording.